dorsal-ventral axis of dentate gyrus, which was studied from Hen's labs, and this was published in Neuron on March 2013. Before going through this study, let me briefly introduce you of a functional distinct structure in hippocampus. Hippocampus can be divided into separate regions, which is rostral dorsal region, intermediate region, and caudal ventral region. Dorsal hippocampus is involved in cognitive function, such as learning, memory, navigation, and exploration. And ventral hippocampus is related to stress, emotion, and affect, and this dysfunction can be lead to psychopathology. And there are supporting evidence showing that gene expression in dorsal hippocampus correlates to the region that's related to information processing, and gene expression in ventral hippocampus correlates to region related to stress and emotion.
Is there markers for better CA1 or
what?
Is there markers or markers? What is that? How they plot? How they start to?
Oh, this is several different genes.
Is there any function along with intermediate?
Intermediate region is like overlap region between dorsal and ventral hippocampus.
How they marked? Is that which genes?
Oh, there are many different genes, but I didn't bring them.
So this kind of situation, right? Which part is pronoun?
Here.
DG. Which part is ventral region?
Oh, I'm explaining about the separate region of hippocampus, and for the DG, this region is considered as dorsal, and here considered as ventral. No?
Which one is ventral? I think this section is ventral.
Yeah.
That section is just ventral.
Anterior part called the dorsal, and the posterior part. This is Encelos paper, right?
Yeah.
To my memory, they said like that, or I'm not sure.
I'll check.
Yeah.
Let me briefly introduce neuronal connectivity of the dorsal hippocampus. Dorsal CA1 has the greatest density and selectivity of place cells that codes spatial location and sense massive and sequential multisynaptic and fit forward excitatory projections to dorsal part of subicular and pre-subicular and post-subicular. And this dorsal part of hippocampal and subicular complex is consisted of head direction cells, which code head position in space. And another predominant projections from dorsal CA1 and dorsal part of subicular complex are retrosplenial and anterior cingulated cortical area, which is involved with cognitive processing, including visuospatial information, memory processing, and spatial navigation. In short, this dorsal part of hippocampus and dorsal part of subicular complex and retrosplenial and anterior cingulate cortical areas mediate cognitive process such as learning, memory, navigation, and exploration. And this is neuronal connectivity of the ventral hippocampus. Ventral CA1 and ventral part of subicular and amygdala and medial prefrontal cortical structures forms these structures forms connection directly or indirectly through lateral septum and medial central amygdala nuclei and bed nuclei of stria terminal to innervate into hypothalamus, which controls neuroendocrine autonomic somatic motor activity, which is associated with motivated behaviors, such as ingestion, reproduction, and defense. So the connection in ventral hippocampus is an ideal situation to regulate the impact of emotional experiences and to control general affective state. In this study, they tried to evaluate the effects of acutely increasing or decreasing activity in the dentate gyrus on cognitive and emotional behaviors. And they focused on dentate gyrus based on multiple lines of evidence supporting that it is effective processing region to be manipulated. And they chose optogenetic to manipulate dentate gyrus with high temporal resolution and cell type precision. So here, dorsal and ventral dentate gyrus cranial cells were optically targeted to evaluate the effects of acutely increasing or decreasing activity in dentate gyrus in awake behaving animals. So they, in this paper, they used PUMC-BET pre-recombinase line to express the target opsins of cranial cells in dentate gyrus. So, and these lines were crossed with conditional mouse lines, yellow light activated holorhodopsin or blue light activated channelrhodopsin. So for in PUMC holorhodopsin mice, they showed selective membrane expression in cranial cells along entire dorsal-ventral axis of the dentate gyrus. And from whole cell recruiting of brain slice from PUMC holorhodopsin mice show robust hyperpolarization, which blocks action potential generation with constant yellow light stimulation. And to conform the functional impact of holorhodopsin activation, they exposed mouse to the novel context with yellow light illumination, suppressing the cranial cell activity in dentate gyrus, where the optic fiber was implanted. And they had confirmed that PUMC holorhodopsin mice show decreased CFOS positive cell induction at implanted region. And for PUMC channelrhodopsin mice, they also showed selective membrane expression in cranial cells along the entire dorsal-ventral dentate gyrus axis. And from whole cell recruiting, holorhodopsin mice showed robust in-order current, which was sufficient to bring cranial cells to action potential threshold with blue light illumination. And they had also confirmed increased CFOS positive cells at implanted site region with PUMC channelrhodopsin mice. And they also confirmed a local.
I have a question. How many areas are covered by blue light? How many areas are covered when they? How many areas are covered by blue light when they do the experiment like that?
The area?
Yeah.
Actually, the diameter is commonly used 200 to 300 micrometer diameter, and
So
the numbering of aperture is like 0.37, so it's like a shape like cone.
yeah.
So
The region I risk is
I think about half of the DG should be covered.
half of the region I risk is POMC is also expressed in the hypothalamus or the brain region, so they're just to be related to each region.
But I don't know. That depends on the power of the distribution.
And they also conform local suppression and activation with dorsal and ventral dentate gyres. So with POMC haloreducin mice, they have shown that when optic fiber was implanted in dorsal region of dentate gyres, the C-FOS positive cells were robustly decreased in dorsal region, and when it was implanted to the ventral region showed decreased induction of C-FOS positive cells. And as you can see in this figure C and G, channel reducin mice showed increased C-FOS induction rostral and caudal to the region where optic fiber was implanted. And this could be due to the recurrent excitatory connection within dentate gyres. But however, they had conformed increased C-FOS induction in CA3 and observed that it was locally specific to dorsal and ventral, so indicating that specific region projecting from DG to CA3. So illumination of the dentate gyres in POMC haloreducin and POMC channel reducin mice in vivo modulates C-FOS levels in hippocampus in a region-specific manner. Okay. So first, they wanted to evaluate whether dorsal dentate gyres had any contribution to encoding and retrieval of contextual fear memory. So first, they implanted optic fibers to dorsal dentate gyres, and they exposed mouse to conditioning context for 3 minutes prior to the single shock of 0.75 milliampere, and with light on, which suppressed the activity of cranial cells in dorsal region. And then 24 hours later, they re-exposed mouse to the same context with absence of light, and they found robustly decreased freezing level of POMC haloreducin in mice, which shows inhibiting the dorsal dentate gyrus cranial cells impair encoding of contextual fear memory. And when they were tested to see whether retrieval was also involved in dorsal dentate gyrus, but there was no difference altered between two groups. So they had confirmed that specific role of dorsal dentate gyrus in rapid encoding, but not in the retrieval of contextual fear memory. And then they moved on to test whether ventral dentate gyrus was involved in encoding and retrieval of contextual fear memory. And they had found no differences between two groups. So they had confirmed that inhibition of cranial cells in ventral dentate gyrus impaired neither encoding nor the retrieval of contextual fear memory. So dorsal dentate gyrus had selective role on encoding and retrieval of contextual fear memory. Then they wanted to test whether dorsal dentate gyrus was involved in spatial learning by using active place avoidance test, which has shown to be sensitive to dentate gyrus manipulation. So let me briefly introduce of this test. In active place avoidance test, mice are placed on the 40 centimeter diameter circular platform, which rotates clockwise at the speed of 1 rate per minute. And the circular platform is placed in the room environment with multiple visual cues. In this paper, they have used black curtain and a white cardboard. And shock zone is defined within 60 degrees in circular platform. So when mice enters the shock zone, they receive a foot shock of 0.2 milliampere for 500 milliseconds. And if they stay in the shock zone, they receive the same intensity and duration of the foot shock for every 1.5 seconds. So basically, mouse has to learn to avoid the shock zone. So for the
What the 60 hertz implicating? Do they give us
tone. Okay. For training trials, they conduct 3 sessions. Each section runs for 30 minutes. So session 1 with light on, session 2 with light off, and session 3 with light on. And after 24 hours in conflict trials, shock zone switched to the opposite quadrants, and mice are tested for 20 minutes with light on.
Mice are freely moving, right?
Yes. So in active place avoidance task, optic fiber implanted mice were placed on the circular platform and trained to avoid the shock zone, while their dorsal dentate gyres were suppressed by constant yellow light. And they had confirmed that optogenetic inhibition did not affect exploration or acquisition or retrieval of spatial memory. But when the shock zone was switched to the opposite quadrant during conflict trials, mice had shown robust deficits. Ponzi holorhodopsin mice entered the new shock zone significantly more compared to the control and stayed less time in the safe zone. So they had confirmed that dorsal dentate gyrus cranial cells modulate cognitive flexibility in the resolution of conflicting memories.
Is there any another paper experiment that which says that dorsal dentate gyrus is involved in cognitive flexibility, or not? Using another paper experiment? What kind of paper experiment? You didn't have?
I haven't seen it. Spatial theories might be the one.
Dentate gyrus is involved.
Not specifically the dorsal dentate gyrus, but
Specific, but also dentate gyrus is involved now. Why do you use the kind of experiment? That is not commonly used paper.
yeah, this
experiment.
is very rare. But yeah, they found the difference between two groups. And then they wanted to see, they had confirmed that suppressing dorsal dentate gyrus impaired encoding of contextual fear memory, but not retrieval. And suppressing ventral dentate gyrus had no effect on either encoding or retrieval. So they wanted to see whether elevating the dentate gyrus in a region-specific manner would have any effect on encoding or retrieval of contextual fear learning. So first, they implanted optic fiber to dorsal dentate gyrus and see whether activating cranial cells in dorsal dentate gyrus had any effect on contextual fear memory. And they have found that the freezing level on contextual fear learning, on encoding of contextual conditioning reduced and retrieval also reduced. So they have confirmed that elevation of dorsal dentate gyrus selectively impairs the encoding and retrieval of a hippocampus-dependent contextual fear memory. And they also had confirmed that the memory form didn't erase. And then they went on to test whether activating ventral dentate gyrus had any effect on encoding and retrieval of contextual fear memory, and they found they had impact neither encoding nor interval of contextual fair memory, so which conforms that specific contribution of the dorsal dentate gyrus to contextual encoding.
And this graph, the phrasal level of the control good seems a bit high. It's more than
Yeah.
80%. So do you have any?
Overall, they showed high freezing level, and
Do you get two shocks?
Huh?
Do you get two shocks?
and
Seriously.
the shock they received was pretty high, 0.75 milliampere. That's pretty high for shocks, so that
That
might
makes
be.
sense.
But I remember that Eun-Hae 박사님 told us that if
Dr. Lee.
Dr. Lee told us that if the freezing levels go above 60%, that might be the saturation could be happened, but because freezing level decreased, it's reasonable to.
Previous that. Next. Are you talking about next? Are you talking about this graph, right? Yes, I asked about the decrease of little bit decrease in retrieval test, right? No, no, it's increase of the.
They decreased, but not significantly.
Yeah, but I think that could be the later result shows the possibility
Oh.
of that kind of decrease.
And because the number of mice they use is not that
No? How many miles?
6 to 7 or 5 to 6. So yeah, so that might be the reason for that. And finally, they wanted to see whether dorsal dentate gyrus or ventral dentate gyrus had any contribution to innate anxiety-related behavior. So they chose two well-validated tests, EPM and elevated plus maze and open field test. According to innate aversion to open space and bright space, mice avoid open arm of the EPM and center of OFT. So first they inhibited dorsal dentate gyrus and ventral dentate gyrus. Then they found no differences between two groups through EPM and OFT. So they confirmed that inhibiting dorsal dentate gyrus cranial cells or ventral dentate gyrus cranial cell has no impact on innate anxiety-related behaviors. Then they elevated dorsal dentate gyrus cranial cells, and they had found very interesting results. So when they activated cranial cells in dorsal dentate gyrus, they had found robustly increased exploration in a novel context. And in EPM, POMC channel rhodopsin stayed significantly more time in open arm when the light was illuminated, but there was no differences in open field tests. So, yeah, so they had concluded that activating dorsal dentate gyrus only increased exploration behavior.
It is affecting after light off state, right? So
Yes.
when they shed the light on the place, the exploration, the
What?
total distance moved was increased,
Mhm.
but even though they turned off the light,
Oh,
still.
yeah, because that increased exploration times is still if natural lights off. This they explain that this could be due to the plasticity of mostly fiber and CA3 synapses as a consequence of stimulation, and or another reason could be due to a decreased aversion to the set task apparatus, because it's not novel anymore. And usually OFT takes only 5 minutes, but in this case the total duration was 15 minutes, so that could be the reason why.
Is there any reason you used the 50-minute for here?
EPM?
The 15 minutes?
Oh, because they have to test light on off.
Control group shows. The control group shows the decrease of testing. So that's not the.
As Eun-Hae said, it could be gradually. I mean, it is not normal. I mean, it is not novel anymore in the context. So it could be why they gradually decrease.
They feel boring. They feel bored.
But overall, channel products in mice explore significantly more, even in light on.
I believe this is not specific because
Not
previously the hallucination experiment they didn't show any effect, right? So that region is really involved. Well, I think they should do some other experiment like they give some anxiety situation, some nervous, then make them nervous, then give hard option that if they became calm down or some reduce the locomotion, then we can say this is specific for that behavior. I think because the I1 channel motion mice also showed the increase of showed people increase of the activity and movement after light activation. So this could be some other effect, I believe.
maybe.
But their expression is just related to granules. Granules are.
Yeah, but however, there is a possibility. The hippocampus is so huge, huge complex structure.
I think it is okay to say they increase the anxiety-like behavior, right? So, because OFT and EPM is only the test that can only test innate fear or innate anxiety. So, if we want to test another anxiety test, that could be better, but
Yeah, but I want to say is that could be more beyond the
Yeah, I
activity
think
of
further,
lesion.
if they do some additional experiment, this phenomenon will be more clear. And when they moved on to elevate ventral dentate gyrus of cranial cells activity, and they have found that their anxiolytic behavior was robustly reversed with light illumination, which activates cranial cells and ventral dentate gyrus. In EPM, the mice spend significantly more time in the open arm, and OFT mice spend significantly more time in the center area. And their total exploration did not differ between two groups. So they had concluded that dorsal modulates exploration via dorsal pole and anxiety via its ventral pole.
In this case, there is no. Their increased locomotion is just restricted to the light on phase, and when light is off, there is no more changes in behavior.
So I don't know why that happened.
We never know.
So I think they are guessing that it is more to do with the exploration with dorsal pole. So, as a summary, they had confirmed that by inhibiting dorsal dentate gyrus, they had confirmed that dorsal dentate gyrus is required for rapid encoding of contextual information, but not for memory retrieval. And by inhibiting ventral dentate gyrus, they had confirmed that ventral dentate gyrus is not required for contextual fear learning. And by elevating dorsal dentate gyrus, activity induced dramatic increase in exploratory behavior in the novel environment specific. And ventral dentate gyrus exert influence on innate anxiety-like behavior. And these results altogether can point to that dorsal ventral pore of hippocampus are functionally distinct, so that hippocampal activity not only has mnemonic function, but also strongly influence anxiety-related behaviors. And this paper had pointed out two interesting results: that activating dorsal dentate gyrus dramatically increasing exploratory behavior, and activating a ventral dentate gyrus robustly decreasing anxiety-related behavior. So this could be explained by that differential behavior effect may be due to driving hippocampal output in a region-specific manner. But they further studies had to be conducted to examine which specific region modulate this specific behavior. So the potential candidate for ventral dentate gyrus is amygdala and prefrontal cortex, and potential candidate for dorsal dentate gyrus is lateral septum, ventral tegmental area. And lastly, by showing that activating ventral dentate gyrus robustly decreased the anxiety-related behavior, they point out that ventral dentate gyrus might be very reasonable target to treat anxiety with minimal cognitive side effect. So further study also has to be done on this too. Yeah.
I have just one question.
Mm-hmm.
Is there a clear cut that discriminates dorsal and ventral?
I'm not sure, but I think they used the reasonable reason.
And how can we confirm that the right on dorsal did not affect the ventral region?
Because the laser was illuminated region specifically
What?
and very robustly.
They also checked the Shito's expression patterns in previous.
Yeah, and also they showed that local suppression and activation with CFOS induction, positive cell induction. Yeah.
I think it says that if you keep the light in the ventral part, wouldn't that affect
The dorsal
part
part.
as well?
I have to check. I don't. Might be, but we're assuming. But I don't think illuminating laser to while illuminating laser to the dorsal part would affect the ventral, because I believe it's region specific, and it turns on and off very robustly.
Is it possible if you keep the light on the ventral, is it only going to be ventral part, not affecting dorsal?
Sure. Because if we use
why they use tangerine mouse? They can just express virus in the.
I think that's convenient and strong phenomenon because people there are not many papers from hippocampus using orthodontics. I think that the effect, it is not easy to see effect from hippocampus.
It's broad
Broad and large, and there are so many compensation, I think. So to see the effect, I believe tension is much easier and stronger.
but it's not specific.
Yeah, a little bit overlap.
The result of this paper is pretty straightforward. Thank you. I have one more slide.